my name is Brice Adams. I am with the Conservation Genetics Group up at Abernathy. And most of the work that I've done since I've been with the Fish and Wildlife Service has mostly been the fisheries perspective. Bull trout in the Clark Fork system, Olympic mud minnows, the endemic species found in the state of Washington, Oregon chub, things like that. And luckily, a couple of years ago, we were given this opportunity to sort of branch out and work with what we sort of jokingly refer to as the greenest member of fisheries. And that's this plant species, the Spalding's Catchfly. And so it's a medium herbaceous perennial, so it's about 8 to 24 inches when it's mature and standing upright in the field. And it can be found mostly in grassland habitat, sagebrush steppe, open pine forests. And in this case specifically, you find it in eastern Washington, northeastern Oregon, western Idaho, and sort of northwest Montana. And when botanists were looking at this species, they sort of identified 5 major physiographic groupings. So sort of how they thought, based on the habitat and various other factors, sort of how these plants should sort of be grouped and managed. And in this case, so we have the scablands, which is sort of this large chunk. This map was taken out of the recovery plan. So it may be a little bit more difficult to see here in the audience. Those at home might have a little bit better perspective on this. But we have the group here sort of in eastern Washington. This is the scablands. We have the Palouse sort of interchecked here. Canyon grasslands sort of in the border region between the 3 states. Blue Mountains found in northeastern Oregon. And then the intermontane, which is this group of populations found in northwest Montana. So this species was listed as threatened in 2001. And what I have here are just a subset of populations that we know of and their census size that they knew of at the time of listing. And so we have a pretty wide variance. We have some populations that are as few as potentially 60 to 62 individuals. And then you look down here at Zumwalt Prairie, which is a plot of land owned by the Nature Conservancy, and we have maybe upwards of 10,000 of these Spalding's Catchfly in this habitat. So the question is, how did we get where we got? This is kind of the same story you'd probably see with a number of organisms, not only just out west, but also in the United States, is we had the impact based on development, land type changes. What was, of course, a native prairie is now suddenly 10,000 homes or something like that. So the habitat was taken directly away, and so that this species is no longer there anymore. Next, of course, agriculture. Either traditional farming or cattle in the same habitat. That's going to have an impact on this species, either it be trampled or eaten or something like that. And of course, invasive species. In this case, we have the yellow star thistle and the spotted knapweed. These were species that were not found in these same habitats. And based on human movement and a few other things, these have now entered that same habitat with the catchfly and have a direct impact. Either they are using the same resources or they're just out competing. And of course, fire regimes. We've done a number of things, both changing the intensity and the frequency of fires. And of course, that's going to play a role in what species we see in these habitats. And finally, it's maybe a little bit, again, more difficult to see here in the audience, but what you see here are all these little black dots are known populations of Spalding Catchfly. And the big picture you can take from this is that there's big spaces between a lot of these populations. And so this is an organism that's pollinated by mostly bees and a few other organisms like that. And if you have large gaps between these populations, it's just going to be more difficult to have gene flow have anything going on, just because there's large stretches that those pollinators have to go through to find that next population. So with that in mind, when the species was listed, of course, one of the first things they started working on is, what are the recovery goals? What are we going to do to get this species off the endangered species list? And so some of the major things, of course, is conserve what we currently have. Can we identify other populations that maybe weren't known at the initial time of listing? If not, can we develop potentially new populations, or can we expand some of these populations that we know of that might be limited in some other way? And so with that, they want to establish 27 populations throughout the species range that had at least 500 individuals. And that 500 individuals is the census number. So they wanted to go out there and see if at least 500 plants in each of these populations. And of course, to see stable or increasing numbers in those populations, and I think the number is over 15 or 20 years. So the purpose of our study was to look at the genetics of this organism. There hadn't been a previous genetic study of this species, so we were able to go through and identify some new markers that would hopefully be informative for us. And using those, we were able to try to look at what is the genetic variation that you see not only within those potential physiographic groups, but across the range as a whole. And so what we see here is those 5 physiographic groups again, but in this case I've added some color to them, so hopefully they can pop out a little easier for y'all. And then we have the 19 populations that we sampled. Of course, these are not all the populations that exist. These are the populations that were most easy to get to, had large enough numbers that we could sample from, things like that. But what we were able to do is get a number of populations from each of those physiographic groups so that we could sort of get an idea on the broader perspective for this species. The next thing we were hoping to look at is, could we identify individual populations that maybe had a low amount of genetic diversity from those 19 populations that we were able to sample? And if so, is there potential for genetic enhancement? Could we bring in other individual plants to hopefully add some genetic diversity to that population? And so I just have a quick little figure to sort of explain what I'm talking about. So we have theoretical population A, which has a large number of plants, and population B, which might only have a few plants in it. Now looking at, of course, you might look at population B and say, there's something wrong with it, we need to do something about that. But the first thing we need to look at is, what is the genetic diversity in population A, and what is the genetic diversity in population B? If there's high genetic diversity in population B, yes, we might want to move some individuals in, but it might not be, we need to do this immediately, that there are dire consequences. So after we know that B has lower genetic diversity, the next thing is, do they have a similar genetic heritage? Can we move plants in from A and it won't have a negative impact by outbreeding or something like that on population B? So after we figure that out, then we can actually potentially move plants over from A to B. So it's just sort of setting the table before we actually just go out, pick up plants, and move them into the next population. So the first thing that happened is we had to have some wonderful people go out and collect specimens for us. And in this case, they sent us leaf specimens, a handful of leaves from each individual plant. They came fresh, they came dried, and then we got to grind them up using this wonderful machine. And then this fine little powder in the end is what each one of those leaves looks like. Mostly that just gives us a greater opportunity to get DNA out of the plants. So as I said, we were able to identify 10 genetic markers to look at this species. We had 498 samples overall across those 19 populations that I showed earlier, and had about 26 plants per population. So we figured we had a good number to sort of have a good idea of what the diversity in that potential population is. So the results here, you have the 19 populations, and they're color-coded based on those physiographic regions that I was showing earlier. And sort of the takeaways from this figure, the first is this column here in the middle. This is heterozygosity. So as a geneticist, this is our way to sort of look at what is the genetic diversity that is in a population. And in this case, of course, green, higher genetic diversity, red, lower genetic diversity. So what you see is we have kind of a lot of variance across all these populations. We see that the intermontane, so remember those are those populations sort of in that upper corner of northwest Montana, have lower genetic diversity than most of the rest of the range. We also have a few other populations that are showing kind of low genetic diversity. The next thing to take away is we have two columns here. Column on the left is the census size. Again, this is the biologist going out saying, hey, how many plants do we have out here? Well, the next column, this is the effective population size. So this is kind of the genetics way to look at how many plants are potentially contributing to that population. And the big takeaway from this is that the number on the right, the genetic number, is much lower for most cases than the number on the left. So in other words, not all plants that they can see out there are contributing equally as all the others. So that number, that 500 number that originally what they were looking at might not be a great way to mirror on how a population is doing. The next thing we looked at is what is the structure of this species. And so we looked at a principal component analysis. I don't actually have it up here. A lot of people sometimes look at PCAs and just their eyes glaze over. So I just kept it a little simpler and we just put up some maps. So these are those 19 populations I was talking about originally and color code based on those 5 physiographic groups that were the original designations for these populations. And what we saw after the PCA is that the physiographic groups are probably not a great mirror for what the actual genetic structure of this species is. That we probably have sort of a main range genetic structure. Most of those populations could sort of be managed as one larger group. But we found interestingly enough, this set of populations kind of down in the Salmon River in Idaho that seemed to be differentiated from populations that were just a few miles away. And so they started to kind of pull out from everything else. And then those intermontane groups might potentially be split up into 2 management groups. So the next thing we looked at is a structure plot. Many of you may have seen these, but if not, I'll just give a really brief explanation. So each one of these is an individual plant. There's an individual bar in here. And what the color codes are telling us is what its proportional relationship to a potential grouping. So in this case, we had 4 potential groupings. And what you see is for the most part, most of the individual plants were assigned to a single group. And again, this is the map showing the original 5 physiographic groups that we saw the same thing, that the genetic structure in this species probably doesn't match up with that original physiographic grouping that was established for the species. So the markers we were using are great at telling us sort of the short term, the last, 50 years, something like that structure of this species. But we wanted to look at what is potentially the longer term structure. Does this picture match up with what has potentially been there in the longer scale? So we looked at chloroplast, that's just like the mitochondria that we all have. So it's something that's going to be highly conserved, so there's not going to be a lot of changes, so it should help be able to tell us these answers. So fewer individuals, but again, all 19 populations. And again, the big takeaway from this is that those Salmon River populations that we noticed with the microsatellite, with the other genetic markers, were popping out with this longer term scale, telling us that there has been some kind of differentiating event that happened a long time ago and made these group of populations relatively unique for a long period of time. So the question is, what are the long-term management implications for this species based on the genetic information that we're able to hand over? And so taking those original recovery goals that were established, the 27 populations across those 5 physiographic groups, sort of what you see imposed here in the red numbers are what the original plan was, that we'd have 8 individuals up, 8 populations up here, 4 in the intermontane, et cetera. But because we're showing that the structure of this species probably doesn't match up with this physiographic groups, they're able to potentially change the populations that they're going to be able to put their most energy into in conserving. And these populations down the Salmon River, which might have been ignored previously because they're very close to other populations, can now suddenly have a special conservation value that, like I said, may not have been known without genetics. The next thing, again, that second point of, can we identify populations that have potentially low diversity? And we were able to do that. What we have down here is Lost Trail National Wildlife Refuge. We have a small population of the plants there and had the lowest genetic diversity that we saw. So the question is, they're looking into potentially going to Dancing Prairie, which is the population up here on top, and moving potential plants down into Lost Trail to kind of give it that genetic boost that we were talking about. So a really quick summary, since I'm running short on time, is we have a species here that was listed as threatened in 2001. Due to the work on this project, we were able to identify some new genetic markers that can be used for all future analysis on this species. We saw that those original physiographic regions are probably not very representative of the actual structure of this organism. And just simply looking at the number of plants that you see in the habitat might not be a great indicator on the actual number of plants that are contributing. And so maybe that 500 number originally established might more be reflected based on the genetic number as opposed to maybe the census number. And so with that, I have to thank the Boise U.S. Fish and Wildlife Service office for the funding of this project, and of course the large number of contributors that went out to collect specimens, but also were integral in with the analysis. So with that, I will take any questions. So the question was about those Salmon River populations. Was it taken in the wilderness area or was it taken across? So in this case, it was, I don't believe it was in wilderness area. I believe it was just Forest Service habitat for the most part. I'd have to go back and look at the map. But in this case, this was the 3 largest populations that we could find in that immediate area. But you're right, it does look like there's kind of been this long history of divergence. So.